guys, in today's video we're going to talk about replication in prokaryotes, but using a fictitious analogy of Candyland and gingerbread scientists. As we speak, there are currently gingerbread people stuck on the sides of the mountain trying to get out. One of the scientists gets an idea to use double-stranded candy cane or DNA from the sky and separate its strands into two pieces to replicate, given it is replicating in this direction. Immediately they get to work and use a nearby gyrase tool to release supercoiling of the candy cane DNA ahead of the replication fork. They also use a helicase tool that attaches to the top of the mountain and breaks into the hydrogen bonds, holding the candy cane DNA strands together as it moves through the mountain replication fork. To ensure that the candy cane DNA does not loop into hairpins, they use large nail, single-stranded binding proteins as a stapling device. A gingerbread person was confused on which side of the mountain would the DNA be a template strand, so the head gingerbread scientist explained that the template strand would be on the left side of the mountain because its leading continuous strand would code in the 5' to 3' direction with the mountain replication fork. The right side of the mountain would be the coding strand because the replicated material would be building the lagging strand or Okazaki fragments in the opposite direction of the replication fork and would require primers. It is important to remember DNA is added to the free 3' OH end. Moving on, the next tool is the primase tool that auto-generates RNA primers that bind at the 3' end of the candy cane strand and act as a binding site for the start of replication. The gingerbread people then use the DNA polymerase III tool to add nucleotides to the existing primer and grow a new candy cane DNA replication strand. Another similar tool is the DNA polymerase I tool that will proofread and remove primers in the 3 to 5 or 5 to 3 directions and fill in the nick or gap in between the DNA strands, the lagging strands. In the case that the DNA 5' to 3' exonuclease activity malfunctions, the primers will not be removed and thus cannot be replaced with a nucleotide base. Moreover, DNA ligase cannot come in and seal the nick in between the start and end of Okazaki fragments as it usually does. This would result in the fragmented DNA that would be non-functional. Finally, the last tool the gingerbread people use before they can escape the mountain is telomerase that makes repeats of the candy cane to extend the 3' end of the telomere to prevent nearby candy cane fusing or deletion of material.